Thank you again, Jacob, for being willing to speak. Now we'll move on to Gary Ann's talk. That's a lot of topics to cover in a short talk. So
yeah, so it's actually going to be mostly on the first part. So I can't start sharing the screen while someone else is sharing. There we go. Okay, so the title is a mouthful. Okay, all right. Yeah, so a mouthful of the title. Just for those of you who don't know me, I'm actually a clinician. I'm a surgeon, critical care physician. You know, so I've been doing a lot of this computational stuff, but a lot of it is focused on trying to improve kind of what we're doing in the clinic. May not necessarily be directly related, but in general, I have this very translational bent. So that kind of is a bit of a framing for what I'm wanting to talk about. So, you know, obviously we're dealing with COVID right now, and, you know, I'm probably not alone in having some frustration about the relative limited number of things that we could do to try and mitigate, you know, the response to the pandemic. Basically, we're stuck with, you know, public health, non-pharmaceutical interventions, which, you know, of course, are nice, but there's a, you know, a huge challenge with respect to what to actually do in terms of people who get sick. You know, the management of the sickest patients has, you know, significantly improved as people learned how to take care of COVID-related respiratory failure. The steroids is definitely, I think, a positive. And then there's an entire alphabet soup of cytokine type mediators that are out that is one of the things I want to talk about. So, you know, this challenge number one is the limitations of what we can do. The first one I mentioned about. The second one is coming up with novel antivirals is really, really hard, because coming up with antivirals is really, really hard. And vaccines take time. And, you know, the companies that are rolling out the vaccines now have done, you know, incredible work from that standpoint, but it is essentially a, you know, a time-consuming process. So the challenge in my mind is what can we do between, you know, the public health, non-pharmaceutical interventions and vaccines. And we saw a lot of repurposing of existing drugs. How would they work? It was essentially done empirically. In many circumstances, a very haphazard way that led to a lot of confusion and, I think, misinformation with respect to what could be done. I think in preparing for how to actually treat some of these patients, gain some sort of insight into the pathophysiology of candidate pandemic viruses, right, which is a comparative virology zoonotic reservoir challenge. And then shorten this time to effective vaccines, which is accelerating the pipeline. We'll talk about that. Right, so my proposal, and hopefully is shared within this working group, is that the responses to challenge number two can be accomplished by building a repository and infrastructure for multi-scale modeling to aid in these particular processes, these particular steps. Right, so just a little bit of my modeling credibility here. These are my primary focus has been, as Jacob mentioned, in bacterial sepsis, systemic inflammation. This innate immune response, ABM, is a model that's, you know, about 15 years old, 20 years old now. We have, you know, multi tissue models. So that was just
sorry, your screen black, Gary.
Okay, I'll stop that one. And then we repurposed a existing model of, oh, that one died too. All right, so it doesn't like me running movies. That's okay.
At least me, I'm only seeing the whole screen black.
Okay, oh. Hang on a second. It's these movies have occupied the bandwidth. Let me go back.
Yeah, it's back.
Okay. And then I'm going to focus talking about this innate immune response agent-based model, which we've done in silico trials of multi-mediator therapy of the type that have been tried with respect to the COVID cytokine storm. And we'll jump off that in a second. Our current model that we're working on is the cellular immunity agent-based model, which is essentially the innate immune response model with the cellular immune components added into it. Here's a wireframe diagram of that particular model, shows some of the various components associated. It's obviously pretty complicated. I think there's been some discussion previously about, you know, simpler versus more complicated models, and the challenge of parameterizing these types of things, which is exactly the problem. And that's what's going to form the bulk of this talk. I would contend that, that if you're going to try and come up with interventions or want to evaluate existing interventions, you need a model of the same level of resolution so that you are limiting the number of inferences you're making about the effect of that particular intervention. And these particular circulated mediators with the CIABM represent cytokines for which there exist, pharmaceutical grade monoclonal antibodies or antagonists, and probably were tried during the COVID epidemic. So I would contend that in order to evaluate somehow prospectively what combination or when these types of interventions should be applied, having a model of the level of resolution that incorporates the quasi-mechanistic effects of these components is necessary. Right, so this gets to the issue of biological heterogeneity, and this is something that all of us deal with. It's a difference between species, it's a difference between individuals, and importantly, with respect to how we calibrate and ostensibly validate our models, the difference between samples, right, as manifested in the inevitable error bars associated with any sort of data collection. And traditionally, biology is focused on the differences between particular systems, right, with ever, microscopic subspecialization of things. But, this is kind of a recurrent theme, and what I'm interested in is that there must be some supposition of similarity from one system to another that allows you to take knowledge extracted at one level and apply it to another context. Right, and I've written about, how the role of multiscale modeling in crossing this particular translational divide in this little opinion piece in the Bulletin of Mathematical Biology. With this idea of using multiscale models as conserved pieces of knowledge that allow you to contextualize data from one circumstance to another is a crucial point. And, so when I showed you that diagram, the CIABM, each one of those arrows represents a rule. That rule incorporates a whole series of parameters, and we know that there's a huge challenge in the parameterization of these models. And in many circumstances, the goal of many modelers is to try and lock down these parameters as much as possible. With, I think, the negative side effect of creating very brittle, non-scalable, non-generalizable models that miss the richness of the heterogeneity that's present in the biological data, right? Because assuming we don't have measurement error, the thing that we know about a series of data across a set of error bars is that system was able to generate that data. Something about that system was able to produce that. So when I think about at least how we, convert biological knowledge into computational rules for a model like an agent-based model, it follows along this kind of, very simplistic generic pathway representation above. And, while we would write this relationship in a certain sort of way, with an arrow going from one component to another component, the fact of the matter is that there's a whole host of cellular machinery that affects that particular state transition that's not explicitly represented in that particular pathway representation. That could be genetic variation that affects the effectiveness of some of the signaling enzymes or the processing components, or hidden pathways that exercise feedback control that haven't necessarily been characterized. So thinking about it in that sort of way allows us to write that particular pathway with these particular parameters, where the parameters themselves represent this host of uncertainty that we know exists, but we cannot quantify within the context of that particular representation. So if we take all those parameters and all those rules and create a matrix out of that, we essentially have a matrix of the coefficients of all the rules within all the components of that model. And that is something that we call the model rule matrix. I want to just say, I want to thank Chase Cockrell, who's a researcher I work with coming up with this idea and doing a lot of the work that I'm showing here. Right, so we assert that this model rule matrix and the terms within that represents essentially the sum total of the genetic epigenetic variability, unknown regulators on the particular rule structure that we are representing in that particular model. And therefore, this rule form can represent, the rule form can represent what's conserved between one circumstance to another circumstance, be it between individuals or actually species. And it takes into it makes explicit this assumption of similarity that we make whenever we interpret results from one experimental model to another experimental model. So like if we study neutrophil activation in a mouse, we assume that there's something similar with respect to how that occurs in the human being. Right, and my contention is if we want to try and get clinically relevant populations, you need to operate over the entire plausible space of these parameterizations, which is an exponential computational challenge. So the first thing is that how do we refine what this model rule matrix is based on data? And then this goes back to the idea that if you look at a set of experimental data that contains error bars, that's heterogeneity within that, and therefore even the outliers within those particular error bars, again, assuming you don't have measurement error, are somehow reachable by that system that is being sampled. Right, so in many circumstances, when you try and fit parameters, you kind of throw away the outliers, you draw a regression mean through the center of the data, and that washes out a lot of information about how that system can actually function. This is important when you're trying to deal with clinical populations that manifest all that heterogeneity. So what we've done instead is looked at a way to operate across the parameter space represented by the model rule matrix as a means of encompassing data that is actually present, right? So if we go back to the idea that the model rule matrix, the MRM, is a representation of all the genetic, epigenetic, and unknown variability associated with a particular pathway, right? If we operate across that entire thing to find out which combinations can actually produce data that's in the real world, that entire bioplausible space is something that we would say, if we want to design a robust control for that system, we need to operate over that particular system, right? And that avoids trying to focus too much on a particular representation of the model and trying to be more generic. So what we see here in the left lower corner is how the optimization process of fitting the heterogeneous data enriches what the rule matrix looks like. So the rule matrix on the left is inherently sparse because when we write what those rules are, there's only a finite number of interactions that we define. But this demonstrates an example of applying this model rule matrix enhancement to heterogeneous data. What you see on the right here, the range of data generated from a clinical burn population in terms of cytokine time series, and fitting to the heterogeneity of that data, right, or encompassing the heterogeneity of that data provides a much richer optimized rule matrix, which actually looks much more similar to a gene expression profile, right? So that is a kind of the idea that what you're doing is you are enriching what the interactions are within your model by doing this process. Again, the idea is that you're not fitting to a subset, but you're encompassing the range. So as opposed to overfitting, this is actually a robustness aimed process. So once we get this entire huge space, then we need to figure out what are we going to do within that particular space. So The parameter space that we have for when we run our simulations not only includes the coefficients of the rules instantiated in the model rule matrix, but a whole series of external variables that are associated with the perturbation that we're applying. So what is the viral load? What is the virulence of a particular infectious agent? What is the transmission, likelihood, or what is the life cycle of that particular thing? Right, so when we think about a simulation and the parameters associated with that particular simulation, there are the internal parameters, which are essentially the coefficients for the rules, and then the external parameters, which are property of the perturbations. Now, if we're thinking about trying to characterize a clinically relevant region of parameter space, we need to operate over both of these parameter spaces. And that's a extremely computationally intensive, intractable computational task if you try to do it from a brute force standpoint. So we have adopted this nested active learning method, which is a machine learning method that provides a gradient directed search for appropriate characteristics of global parameter space that you're looking for. And this is something that we do to identify the subset of parameters that we will consider to be representative of clinical population that we will then do control discovery on top of using either genetic algorithms or deep reinforcement learning. Right, so a quick sidebar into deep reinforcement learning. Most people know about this through the DeepMind projects, AlphaGo, AlphaStar, et cetera. These are model-based DRLs where there is a simulation model that is the basis for the deep reinforcement learning task. We applied this to the IR-ABM, first circumstance in biomedicine that I'm aware of model-based DRL. Here are some characteristics of how we set up the training process. Without going into too many details, the fact is that we could take a set of conditions with a 46% mortality and come up with an intervention policy manipulating the cytokines within that particular system to make that mortality 0, right? And one of the keys here is this initial project involved simulations with antibiotics because we were simulating bacterial sepsis for which antibiotics are part of the treatment. As I mentioned before, that's not the circumstance with COVID, where we do not actually have an effective anti-infective agent, and therefore we reran our DRL with this particular condition. So again, the limitation here is the IR-ABM is not set up to specifically simulate viral infections, but this is what it would be if you relied purely on the immune system to recover from an infection. So the upper graphs in each one of these panels represents what's happening in terms of the actions that are taken by the AI agent trained from the DRL. And the lower one are with antibiotics. So you can see that there are considerably different policies that the AI agent comes up with, which is not surprising. It also turns out to be a more difficult problem. We take a 75 to 85% mortality cohort and turn it down to 25% mortality, which is not terrible, but it's not the best. And it also shows that the policy controls are more complicated in terms of the non-antibiotic circumstance, represented, like, for instance, the difference between the interferon gamma with the no antibiotics versus with antibiotics. Another thing that's very interesting about this is if you look at the two lower, larger panels, one is effect on interleukin number 1, of which the with antibiotics, did a fair amount of activity with, versus the action on soluble IL-1 receptor, which is an intrinsic antagonist to IL-1. And you come up with very divergent amounts of focus on either one of these two components, the same pathway. So we believe that this is an example of underspecification that's intrinsic to ML and DRL, and that was the subject of a big preprint coming from Google a couple of weeks ago. Because, part of the problem when we do these trainings of these AIs or doing machine learning is we have no idea as to whether or not the convergence is based on the same factors that are present or not. So the difference between the antibiotic and the no antibiotic, focusing on different components of that particular pathway, it's unclear whether or not that actually is a real difference, or is that some sort of stochastic process that came out of the training. All right, so now I'm just going to fly over the three things that we talked about in my title. The first one is this idea of gaining insight about the potential viral pathogens by studying zoonotic transfer. Right, so we know that these zoonotic reservoirs are the source of these novel viral pandemics, and there are extensive research being done in terms of like ecological habitat disruptions that would lead to the transmission of the jumping of potential pandemic viruses into the human populations. I think one of the challenges is identifying what is it that makes that jump actually happen, right? That's an extensive area of discussion as well. It's not unreasonable, however, to think that somehow there might be some insight gained in the pathophysiology of the human disease by looking at what it is the difference between the reservoir and a human, and seeing how a virus with certain capabilities is not lethal to a particular zoonotic reservoir, but then becomes pathogenic in human beings. And this can potentially help predict the manifestation of pathophysiology that would lead to non-antiviral specific, but more host modulation strategies. Right, so I took these two graphs from this Frontiers review on bat immunology, and looked at these two particular areas where bats have dampened interferon response to RNA viruses, and also a dampened response to extracellular DNA or damage-associated molecular patterns. Both of these things lead bats not to have as intensive a immune inflammatory response to the presence of these viruses. It is the increased host response to these viruses that is the basis of the pathophysiologic manifestations that we see in COVID, right? So the question is whether or not knowing more about how these viruses exist within their zoonotic reservoirs can provide us potential understanding into how that disease may manifest in a human being. And I think the hemorrhagic fevers also have similar characteristics like Ebola and the Marburg virus. And then finally, we're going to talk briefly about accelerating vaccine development. Fortunately, Moderna and Pfizer and maybe AstraZeneca have done yeoman's work in getting these viruses to potential delivery we have to all accept, at an unprecedented level of efficiency and potential efficacy, right? However, we can still potentially enhance that particular pathway by looking at how vaccines actually work to begin with, right? To filter candidates and identify systemic and off-target effects, importantly, which are maybe related to some of the vector issues, right? The CIABM that I mentioned earlier was originally developed to evaluate development of cellular and T-cell immunity in the dengue setting. But it goes without saying, to a certain degree, modeling immunity requires modeling infection. And we have, in a very preliminary fashion, looked at matching the CIABM to influenza data. It's not great data, it's in the preprint form, but this is all work under evaluation. I think part of the message that I would hope to get across to this group is that we all become very enamored with our own particular models. I particularly am not assuming that my model is any better than anyone else's model. I think that having a range of these types of things is important. This goes a little bit to Jacob's ensemble idea. And the hope is that this particular working group can help develop a multi-scale modeling repository or resource that can provide biothreat response capabilities across a range of responses, be it going at the public health level in terms of optimizing and targeting interdiction or public health strategies, down to vaccine development. But I think there's very important middle zone of using repurposed, having some idea about what the pathophysiology and the host is going to be, and having some platform in a more rational fashion to kind of shuffle around all these combinations of things that people are just trying empirically. The other thing I would hope to get across in this working group is the importance of embracing the heterogeneity that's present in the biological systems we're studying and the data that's collected, not trying to necessarily wash that away and focus on things like parameter sensitivity analysis or necessarily prediction across an experimental platform, but understanding that necessary translational step into the human population, the clinical population, involves an explosion of heterogeneity with respect to the response capabilities of the individuals doing that. So with that in mind, one of the subgroups that I'm hoping to help lead, this modeling individual disease and response to treatment subgroup, I wanted to try and emphasize some of the development of methods to deal with this particular heterogeneity, provide methodological support to the other groups as they are doing their particular more focused modeling projects, and use those models as use cases for method testing. So I'm just going to finish by saying that I am a guest editor on this Frontiers in Physiology research topic, which is the integration of machine learning and computer simulation to solve complex physiological and medical questions. Ignore the abstract deadline. Basically, the papers are due in April. I would encourage any of you, and I think the MSM community is very well positioned to do this, to send us your work that applies various machine learning techniques either to analyze or to enrich your particular models. And I can tell you that you would get a favorable review, at least from an editorial standpoint, a empathetic review process. So with that, sorry if I ran a little bit over. I'll take any questions that you might have.
Okay. Time maybe for one question. I'm afraid we're on a tight schedule, but Gary talked about so many important issues that this working group could address. I hope if we can't have all of the discussion today online, at least we can start discussion in subsequent and offline channels. Time for one question before we move on.
I can reciprocate and ask a question, but I like your conclusions. But more importantly, you show a slide with mortality. At this point, you lost me a little bit. Can you go back and explain whose mortality of what? Is the virus mortality, the cell mortality? This is why I was confused.
Yeah, simulated patients. So the modeling that I do is operating at a much more under the skin scale than the types of things that many of the things that you're dealing with are looking at, right? So we arbitrarily set a criteria for mortality within the simulations. So 80% of the total system, if that's damaged, we consider that system to be dead. So that is basically the mortality within the context of the simulation experiments. There's no necessarily correlation to any sort of clinical population.
Okay, got it. Thank you.
Okay, thank you very much, Gary. I hope again that there'll be plenty of opportunities. Use the Slack channel and the website and email, of course, to talk. And thank you again, Gary, for covering so many important topics. Next week, we have Amber Smith speaking. We have an open slot if somebody is interested. And then 2 speakers from NIAID on the 17th. And Filippo Castiglione. Then no talks on the Christmas Eve or New Year's Eve. And then in January, continuing with Paul Macklin and Bard Ermentrout. Grace has agreed to speak to us on January 14th. Again, please suggest speakers. They don't have to be working group members. We'd be very grateful.